The light-independent reactions of photosynthesis, also known as the Calvin cycle, can take place both in sunlight or in darkness and result in the creation of energy-rich sugars. These reactions occur in the fluid-filled stroma of the chloroplasts and require carbon dioxide from the air, as well as the ATP and NADPH produced during the light-dependent reactions of photosynthesis. The Calvin cycle begins with the critical process of carbon fixation, which occurs when a carbon dioxide molecule is added to a CO2 acceptor molecule known as ribulose biphosphate, or RUBP, which contains 5 carbon atoms and 2 phosphate groups. This reaction, which only occurs in the presence of an enzyme known as rubisco, produces an unstable 6-carbon product that immediately splits into 2 identical 3-carbon sugar compounds known as 3-PGAs. After this occurs, one of the 3-PGA molecules exits the cycle and will be used to make glucose and other sugars. The other molecule remains in the cycle. First, it reacts with ATP. Then, the product of this reaction receives a hydrogen atom from NADPH, producing NADP and a new carbohydrate compound known as G3P. After that, a complex series of steps known as the regeneration of ribulose takes place that consumes another molecule of ATP. These steps result in the formation of a new ribulose biphosphate molecule, and with that, the Calvin cycle is complete. Now the cycle repeats itself with the fixation of a second molecule of CO2. Note that 1 repetition of the Calvin cycle is required for each atom of carbon that is added or fixed to a carbohydrate molecule. So in order to make 1 molecule of 3-PGA, which contains 3 atoms of carbon, 3 repetitions of the cycle are required. That means the Calvin cycle must be repeated 6 times to make 1 6-carbon glucose molecule from 2 3-PGA molecules. Glucose is the ultimate product of photosynthesis because its chemical bonds store the energy that was captured from sunlight during the light reactions of photosynthesis. And glucose is the key nutrient that is broken down during aerobic respiration to generate the ATP that is used to power the activities of living things.